So it's my pleasure to introduce Mike Norton. He and I were both postdocs at Brandeis back in 2019, 2020, and he's I've always been fascinated by his work on both chemical oscillators in networks and active matter. And so yeah, some of the active matter stuff is what we can talk about today. So without further ado, please take it away, Mike. Thank you.
All right. Thanks a lot, Youngman, for the intro and so much, for the invitation. I'm excited to sort of like meet some new folks through this venue. I guess I'm just gonna like dive right in because it's sort of like a short amount of time. Like Youngman mentioned, I have sort of like two main research interests. One is studying chemical oscillators. The other is studying these like active matter systems. And I'm going to explain a lot more about what's going on in this video on the left shortly. But suffice it to say that I'm studying materials that are sort of like minimal materials that have been built up from reconstituted biological components, right? So researchers are taking one or two or three components out of a living system, putting them in a dish and seeing what happens, right? But the thing about these materials is that they are energy consuming. They're combined with energy consuming enzymes, right? So they're not passive. We're not looking at equilibrium systems. We're looking at systems where sort of energy is being injected into them at the nanoscale, right? And this sort of like begs the question, like, why do we do this, right? And there's like a few different ways you can sort of contextualize this research. And I like to think of it sort of in conversation with, what developmental biologists and biochemists and folks study, right? And so like engineering these reconstituted systems, I think helps provide us a language for understanding life, right? So life is obviously very complicated. There's many more variables and degrees of freedom and parameters with a zebrafish embryo than there is with this microtubule system that I'll be talking about later. But still, we can get some traction, right, by studying these simple systems where we have experimental control over the parameters. But maybe from a materials science point of view, right, if our goal is to try to build new kinds of materials or robotics that are not based on hard materials, right? Life shows us what's possible for those kinds of materials. So I think it's sort of a two-way street. We can talk about engineering, and we can also talk about sort of universal properties of these like far from equilibrium materials. And I guess for this talk, I'm kind of in this bottom camp where I see life as setting the bar that we should try to hit by like engineering materials. Okay, so just a little bit more about that video that we were watching on the left. So I'm calling this an internally driven microtubule suspension. What does that mean? It's a system of microtubule polymers that have been sort of recapitulated from living cells. And all of those polymers are about think of them as like little like rigid sticks that are like a micron long, and they've been combined with this construct of kinesin motor proteins and streptavidin. And so these kinesin motors naturally, in vivo would walk along those microtubules. And by combining them into these like multi-protein complexes, you're able to create relative motion between neighboring microtubules, right? So if you imagine you have sort of two microtubules with a motor protein in between them, it's walking in one direction along one microtubule, it's walking in the other direction along the other. You'll get this sort of extensile motion between the microtubules. And when you put that in with a whole bunch of microtubules and a whole bunch of motor proteins, this is what you get. And I should say also that the system is quasi 2D in the sense that it's been sedimented onto an oil-water interface. So we're really like seeing kind of like the whole material, which is nice, for experiments. To get maybe a better sense of that like extensile motion I was talking about, a collaborator did these like really great photo bleaching experiments. So all of the microtubules are labeled fluorescently here, but if you shine a really bright laser light, you can destroy that fluorescent capability, while keeping the activity of the system. And so if we play this forward, we can see that this square extends along the sort of texture direction that you're seeing here, while contracting in the other. And it's a completely volume conserving deformation. Okay. And I guess I don't know what you guys normally do for like questions and stuff. I guess because there's, I'm happy to take sort of like clarifying questions along the way if that's helpful.
Yeah, we'll have people unmute and ask questions. So yeah.
Okay, great. Yeah, and so I guess one thing that you sort of notice about this material, right, is this like striking texture, right? And like what's sort of responsible for generating this particular kind of stretched taffy looking material. And what's happening is that there is what we call a bend instability in the material. So if you start off with all of your microtubules aligned, any like little like fluctuation in that ordering of the material will tend to create flows that make that deformation even larger. And eventually those deformations saturate into what we call topological defects. And I'll be talking about what those defects are shortly. But this is the fundamental idea, is that this system, because it's always trying to extend, has a sort of like intrinsic instability that generates these patterns. Okay. And so I talked about defects previously, but here they are in greater detail. We have two kinds of topological defects that we sort of name according to their topological charge. And you get this charge by doing a winding number calculation. And it's basically a measure of how many times did our orientation field rotate as you proceed around the core of the defect. So at the core is a region where this orientation field really doesn't have any meaning, right? But directly outside it, you can get an orientation, and the number of times that you wind around gives you the topological charge. And so a minus half defect, that as you proceed clockwise around the core of the defect, you make half a rotation counterclockwise. Whereas for the plus half defect, you proceed clockwise around, you rotate half a clockwise turn around. And that's permitted by the fact that we have what we call a nematic field rather than a polar field. And so this is the field that doesn't have a head or a tail. So this is kind of not like a typical vector field that points. This director field points, but it's invariant to a sign flip, right? And we sort of quantify that mathematically using a second order tensor rather than a straightforward vector field. And you can kind of see here just by examination that this is quadratic in our orientation, right? So if I were to go from n to minus n, I wouldn't change q. And that's sort of the feature that enables you to have these half integer defects. Associated with each of these defects are different kinds of fluid flow. The symmetry of the minus half defect makes them, they're still active, but they tend to be more diffusive in nature. Whereas the plus half defects, because of their polar symmetry, tend to propel around and are more active. Okay, so there's been, a really sort of fruitful area of research recently has been to try to control these microtubule active nematics through a variety of means. And I just want to walk you through sort of the brief history of this pursuit. So the left two images, the one on the left and the one in the middle, remember how I mentioned that the microtubules have been suspended down onto like an oil-water interface? Usually that oil is like an isotropic oil, doesn't have any special properties. But if you replace that oil with another liquid crystal that forms some kind of ordering, then that ordering can have an influence on the dynamics of the microtubules. And so the one on the left, the underlying liquid crystal layer has been aligned with a magnetic field, and now the microtubules are having an easier time sort of flowing up and down than they are from left to right. So you get this laning that shows up. A similar thing is happening in the middle, where now you have these circular focal conic domains of a liquid crystal that form. The sort of geometry and topology of those domains can be a little complicated, but the main point is that there's an easy way for the microtubules to move in a hard way, and so they prefer to sort of circulate around those domains. And so now you see like all these vortices that appear, all those vortices sizes correspond to the underlying liquid crystalline order. And then perhaps more straightforwardly, you can also just put this active nematic into a little like microfluidic well to confine it hydrodynamically. And so that's what's happening here on the right. And we explored this in a somewhat recent paper. So this is all like very, it's like very cool, but the limitation is that these control fields are hard to control spatially and temporally, right? So the microfluidic case where you confined it to a well, you can't really change that geometry once you set it. The liquid crystal method for control has some ability to maybe actuate those liquid crystals, but it's still very spotty. But some recent research has created a new kind of construct for the activity where those motor proteins, instead of being bound permanently together by streptavidin, are bound together through this light-activated protein pair, micro and eyelid. And these are proteins that were sort of genetically modified, taken out of plants by Matt Thompson's group at Caltech. But so when you take those proteins, you attach them to the motor proteins, now you have a system with a sort of light activated clutch. Like the motors are always wanting to walk along the microtubules, but they only create stress in the material when they're bound together. And that only happens in the presence of light. Okay, and so here's an example of that working with the active nematic. These are experiments done by Zara. She's a recently graduated student from Brandeis physics department. And on the left is the light that's being applied to the sample, the light level. And you can see that when there is no light being applied, the system essentially is frozen. And it begins to move again once some light is being applied. And the sort of velocity of the system is related to the amount of light that's being applied. So, it's very cool to apply uniform illumination, but really, what we're after is more that sort of spatial temporal control over the system. And so here's that same physical system with the light only being applied to this small circular domain. I guess it's just worth maybe pointing out sort of some of the other work that's been done in this area. There's not a whole lot, but this is work done by Margaret Gardell and Wanda Pablo's group in Chicago. And so they are using, it's not a microtubule system, it's an actin system with different motors, but nonetheless, they are light activated. And it's a little hard to see in these images, but what they're doing is they've created this elbow shape sort of like racetrack, and the defects preferentially sort of follow that track. And so these are just showing some of the trajectories of those plus half defects as they move. So this is where my research comes in. We have all of these experimental tools, especially these light activated tools, where we have the ability to apply spatial temporal control to these materials to try to get them to do things beyond just the chaotic mixing that they were doing before. And this is just a cartoon showing, maybe some of the exotic states that we might be able to drive the system into. But the question is, like, how do we systematically determine what those inputs need to be to like get us to the desired configuration, right? In contrast here, this is just like an ad hoc sort of pattern that's being applied. It seems reasonable that this pattern would give you the defect trajectory you want, but if you want something more complicated, maybe you need a more formalized approach. And so for that, we need some kind of model for the system. And what I'm going to be showing you next is a model-based optimal control for this PDE system. Okay, and so I sort of showed this earlier, but we first need a field that sort of describes the orientation of our microtubules. And so this is borrowing from like liquid crystal theory. We have this second order tensor that describes both the degree of order and the orientation of microtubules. And I'll try to go through this in a way that's helpful. But please, if there's any questions, let me know. We have an evolution equation for Q that includes both hydrodynamic and thermodynamic contributions. So we have convection, we have a flow orientation E. So this is given by the symmetric part of the flow gradient. And so what you should imagine here is that if you have some ideal extensile flow, that the rods will tend to align with the principal axis of that flow. We have rotation, so just being rotated by the vorticity of the flow, that's given by the omega term. And then this free energy term governs the creation and destruction of pneumatic order. And this is important near the defect cores where you have low pneumatic order. And so what's driving the flow in this case? It's governed by momentum conservation in the Stokes limit. So we have our expected Laplacian of U minus grad P, but then we have this active term here. And so what this is basically saying is that we have an active stress that's proportional to our Q tensor. And if alpha is positive, then we have extensile stress. If alpha is negative, then we have sort of the reverse. We have contractile stress. And a way to get kind of a feel for what's going on is you can plot the force field for divergence of Q for idealized director field configurations to sort of get some intuition about what Q is, what divergence of Q is, and all those things.
Just remind me again very quick of the double dot notation.
Yeah, sorry, that's like Q is a second order tensor, so we're doing like a double contraction. So it'd be like in index notation, it would be Qij, Qij.
Thank you.
Does that help? Yeah. Okay, so we throw those equations to, I'm just going to show you some initial finite element simulations without the control being at play to try to give you a sense of what this system does. So there's a natural length scale that emerges from this system that we call the active length scale, and it's sort of a balance between the tendency for the director field to want to relax and become uniform relative to this active stress strength. And roughly, as the activity goes up, this active length scale gets smaller and smaller, and you get more and more defects. So your defect density goes up as you make activity go up. So I'm plotting here the sort of like most important fields from our simulation. On the left is the director field being indicated by these little sticks. And notice that I haven't drawn a head or a tail on these sticks because we have this pneumatic symmetry. I've labeled the plus half defects with the magenta arrows and the minus half defects with the blue triangular symbols. And then on the right is the flow field that's generated by the active stress with the vorticity and direction of the flow field. Okay, and so then the natural question
intuitively,
yeah.
I mean, the pneumatic director field, is that the motion of the microtubules, and the flow field is the motion of the fluid? What's the intuition?
Yeah, that's a great question. So there's only, and this is we're doing a one fluid, what we call like a one fluid model. So there's really only the velocity of the microtubules. There's no distinction between solvent and microtubules. We can argue about whether that's a good assumption. Sometimes it's not. But the orientation field is just the instantaneous orientation of the microtubules. So this is like the if we were to like look at our experiments, right? It's just like the local tangent to our microtubules.
And then the other one would be I guess the velocity of that.
Yeah, just the velocity. And they're not necessarily like going to be, aligned with each other, right?
Right. Okay. Got it. Thanks.
Yeah. And I think it is can be a little challenging to flip between these like really graphical representations and the experiment. I've sometimes tried to plot this differently to make it, because this like lacks the sort of like stringiness of the experiment, but it is there if you were to like integrate along these like little sticks. Okay. So yeah, I think a natural question to ask, right, is like what happens as you change the domain size, like relative to this active length scale. And this is something that we did numerically, but we also did it experimentally. And we find that like as you start to confine the system closer to that active length scale, you'll get this dynamical state that's very robust in simulations. It's less robust experimentally, but for now we're going to focus on the numerical results. But in this case, we have just 2 plus half defects that process indefinitely and at sort of fixed radius around the container, right? So we essentially have like a limit cycle oscillator for this system. And it's very robust in the sense that you can start from many initial conditions and it will always go to either the clockwise or counterclockwise version of the thing that we're looking at. Okay. And so, back to our sort of idea about doing control, rather than choosing sort of arbitrary control goals, I wanted to, as a first pass, try to get a sense of the system by like working with like the native attractors that it had. So I know that because I sort of alluded to this, but I know that if I have, a clockwise solution, then I must also have a counterclockwise solution, right? And this is, me plotting both of those solutions. And my goal is to, try to find an input for the system, a light field input, that varies in space and time, that gets me from one to the other. Okay. And, so, I want to try to find that alpha, that active stress. This is a proxy for the light. It's maybe not exactly a one-to-one mapping between the light intensity that's being applied to the system, but this is, sort of a first order approximation. And so I want to find an alpha that varies in space and time that gets me from one to the other. So I'm going to just apply the sort of standard approach, which is to write a penalty function of some kind that I'm then going to try to minimize with in order to find a control solution. And so there's three main pieces to this penalty function. The first, of course, is that I want my system to do what I want it to do, right? So I'm going to penalize the system for not being in the clockwise state for all time. And I there's probably different ways that you could go about this. I pose this as what's called a tracking problem, where I have a reference solution that varies in time, and I'm always trying to just like match my current state to that table of solutions at all time. In other words, my Q star is changing in time. So I make some norm, quantifying the error between where I'm at currently and where I'd like to be. I have an actuation penalty. So this is a penalty on the amount of light that I'm applying. And there's a few different sort of philosophies around like why you would want to penalize. In our case, it's not like we have like jet fuel that we're trying to like minimize the consumption of or anything like that. But the system does have constraints. There's only so much light that we can apply to the system. And so one way to sort of coax out solutions that don't use too much light is to apply this quadratic penalty. The other thing is that if we accidentally were to apply a whole bunch of light, we might really veer off in state space, right? We might make a bunch of defects that we don't really want, and that might be sort of hard for the control to work. So I've penalized the activity relative to the baseline activity that gives me the attractor that I want, right? So I need some base amount of activity to have these two attractors, and so I'm just penalizing around that level. And then finally, this is partially just for like numerical regularization, but also has I guess a physical reason here is that we can't apply infinitely sharp gradients to the system, and so we just sort of smooth things out by penalizing gradients in the control field as well. So that's the setup, right? We have something that we're trying to do while also like minimizing, while also trying to like not apply huge amounts of activity or really sharp gradients. And of course, we want to do some kind of like gradient descent on this, but we also need to constrain this search to our governing equations, right? And so the natural way to do that is to use Lagrange multipliers. This is what's called Pontryagin's method. Lagrange multipliers are also called the co-state variables or adjoint variables. Go by different names depending on sort of what literature you look at. The important thing about this is that these co-states or Lagrange multipliers are themselves fields that vary in space and time. Okay, and when we do our turn the variational calculus crank on this system by looking for all these optimality conditions, what we end up with is sort of interesting. We have additional partial differential equations that describe the evolution of these adjoint variables. And it's kind of a lot to look at, but I'm showing it just to maybe give you a brief feel for what these PDEs are doing. And the important thing is that the driving force on these PDEs is the state residual. So this is the error between where I am and where I want to be. That information is sort of like getting pumped into these adjoint variables, and these adjoint variables are helping us propagate the errors for the system that we then use to update our active stress field. We basically use an approach called direct adjoint looping. It's really just like fancy gradient descent. So we use this adjoint to sort of back propagate the errors, and that's very analogous to like what's done now in machine learning. I call it adjoint, running the adjoint, but machine learning people will call it back propagation. Same idea. You propagate your errors, you update your active stress, and then you do that until you converge to a solution. It's a pretty like cost-intensive way of doing things, but it's better than doing a completely brute force search, where you are like numerically calculating derivatives of your penalty function with respect to alpha. Okay, so like, does it work? And the answer is yes. So what you're looking at on the top here is the director field evolution. This middle field is the control field that I was trying to find, and the bottom is the flow field. And so you can see that it's like kind of subtle, right? The defects are slowly sort of twisting around by like time 0.72. You have almost this like very symmetric configuration that doesn't really have a handedness. And then by the end, you are now like rotating in the other direction. And so the way to sort of like rationalize the control, right, is I think to look at this like kind of tipping point between these two states, right? We have a nearly symmetric director field with respect to this sort of axis between the two plus half defects. But the active stress field that's being applied has a handedness to it, and so like that is, I think kind of telling us what we need to know about like how this control is working. It's pushing us towards this point of symmetry, and then just kind of pushing us over the edge to fall into the basin of attraction for the other attractor. You can also, sort of rationalize how things are moving by like looking at, oh, like, is the active stress higher on this side of the defect or the other? Oh, that's why it's getting twisted around, so forth. It's a little hard because it's really a whole domain problem. It's kind of hard to break it apart into those pieces. But I guess this is also sort of like the goal of control, is that it is able to like do, help you come up with maybe solutions that are not intuitive. These are just sort of coarse metrics for the progress of the control. So we have a measure of the circulation. We sort of see at what point it became, the handedness of the solution switched. We see that the control effort is like mostly at the beginning of the system, and we see the convergence towards the desired configuration. So we can also just watch a movie of that. So this is our like reference configuration. That's just the desired state just chugging along. On the upper right is the current state of the system. And when it's highlighted in blue, that's when the control has been switched on. So I'm just like running it a little bit before the control is switched on. And you can sort of see the transformation that takes place. So this is, there are, other ways of like interacting with this system. And so this is work that was done at Johns Hopkins that came out actually like right after we published the optimal control work. But it was nice because we had already done their control problem or something very close to it. And so what's going on here is that, remember that this active pneumatic is just floating on an oil-water interface. They've suspended a magnetic disk above this pneumatic layer that they're rotating with a magnetic field, and they're spinning it up to some very high speeds. that flow field then influences the rotation of the microtubules and eventually sort of creates these vortices, around where the disk is. And so the disk is that sort of the bright spot that you're seeing. And you can see that once it starts spinning, you get the microtubules sort of entrained into that flow. And so we had done a very similar control problem where now, instead of creating an active stress distribution, we take the native vorticity of the system and we augment it with an applied vorticity that we allow to vary in space arbitrarily, which is probably not physical. But we get a very similar thing, which is that I think is pretty intuitive. It's like, oh, you want this system to rotate the opposite direction, we'll apply a vertical flow in the direction you want to go, and eventually it will get there. And so that's what happens. You probably did not need optimal control to do this, because we get a sort of spatially complex solution that we may not really need. But it's kind of a nice benchmark just to show that like this framework does generate solutions, for other kinds of flow fields. Oh, sorry, for other kinds of inputs. So I'm going to change gears a little bit in a few slides. Are there any questions about this part so far? Okay. Oh yeah. So I guess maybe to contextualize the optimal control result a bit more, we can maybe think about this system more abstractly as a system that has two limit cycles with this unstable dipolar configuration, sort of separating them, and where you actually have a continuum of dipolar states. All rigid rotations of this middle one are also solutions, right? And we know it's unstable just from numerical exploration. And so like one could like kind of sketch out like a cartoon model that's not a PDE, right? That describes the coarse behavior of this system. And the reason why I think it's helpful to look at this is because I think it illustrates for us in a way what the optimal control solution did for us. And what I mean by that is that in this representation of the system, we've got two limit cycles, and it's obvious that to get from one to the other, I need to apply, a perturbation in Z, right? But in our like higher dimensional system, it's like really unclear like what Z even means, right? Like how do I find that Z coordinate that I want to traverse? And optimal control kind of did that for us. It found the right shape mode, right, that gets us from one attractor to the other, right? Because any other you can think of like arbitrary perturbations to one limit cycle as having two components. You have a component that changes your phase and a component that moves you closer to the other attractor, right? And so our control solution is probably still like a superposition of those two things, but I think it's at least moving us in that direction. I just wanted to plug a recent collaboration, with young men, Michael Chung and Alan Zachariah at UFL. We're trying to do like a data-driven approach to like formally do this model reduction. So the thing I'm plotting here is a cartoon that I just came up with based on the dynamics, but by analyzing the data from the PDE simulation, I think we can actually formally do this model reduction. And so that's just some exciting work that's happening right now. This plot was done by Michael. And I guess I'll just say that like our current goal, right, is to try to combine this in real time with experiments that are going on. Okay, so I guess I only have about 10 minutes left. And I'm just going to like briefly tell you about some recent work where I consider a related problem, which is like, how do we actually design materials that exhibit the patterns that we want, and don't try to steer them there through external means, but design them in such a way that they have the properties you want. And to sort of orient us, we can think about this sort of like schematic for like how the key fields in the nematohydrodynamics interact with each other. We had the Q, which described the orientation of the microtubules. That Q is manipulated by the velocity of the system. But Q changes the sort of the orientation of the active stress that's being imparted into the fluid. So you have this feedback between all these fields. My current interest is around like what kinds of biochemical processes can we add to this system that allow us to change the active stress field in an embodied way, right? And there are sort of a few different aspects of this. I mean, one is you can imagine having a reaction diffusion process that just changes your activity field and that's it. But a problem that I'm interested in, and this is very much inspired by biology, is to think about how does our structure, can we think of a reaction diffusion system that takes as input some aspect of the structure of the material, right? So we have our orientation field. Can that orientation field influence directly a reaction diffusion process? And so it's this problem of what I'm calling sensing, which shows up all over in biology. Biology is able to sense stresses and geometry all over the place, but we really haven't built artificial materials that have that same ability. And just like as an exemplar for like what kind of feedback is out there, we can look at morphogenesis. So the Hydra microorganism is very well studied because its body plan is extremely robust to perturbations. Coincidentally, its body can also sort of be described in terms of the nematic order of this actomyosin network that sort of pervades the organism. So this organism is basically like one big muscle, but there's like some interesting topology stuff going on where we have defects that locate various features of the animal. And one thing that's been proposed is that these defects in this actomyosin network are at least in the mix in terms of helping regulate the body plan of this animal. And what do I mean by regulate? I mean that really large excursions in phase space, in sort of like morphose space of the organism, by like, doing really ghastly things like chopping it up, right? The organism will still regenerate back into a hydra. And so in some sense, the hydra body plan is like a high order dynamical attractor, for this system. And so that's sort of the inspiration for my work. I had asked this question like a few years ago and been thinking about it ever since. It's how do we build like a reaction diffusion system that can maybe sense these topological defects? Can we make a chemical system that's sort of smart enough to find these, kind of complicated features, right? And I think it might be challenging to like really get through this in 5 minutes, but I will just, I'm going to try, I guess. And so what I'll say is that the sort of like hypothetical object that I came up with that can help us do this is what I'm calling a bend-activated reaction dipole, right? And that's like a lot of words, but the idea is that basically we have an enzyme that points, and it points in a particular direction relative to the curvature and bend deformations of the nematic in which it's embedded. And I'm drawing it, here as a kind of a two-headed particle, right, where the products of one side are immediately consumed by the other. But really any kind of directed transport will get you this. And so I think a good example would be like protein pumps, these like ion pumps, any kind of directed transport like along filaments could also like break this symmetry. And I'll just kind of give you a qualitative overview of like how this sort of proposed system works. We note that we have like these areas of like high bend around the topological defects. And you imagine that if you sort of decorate those highly bent areas with these dipoles, that they will like point away from the plus half defects and towards the minus half defects. So we can write a kind of quick field theory that gives us that behavior. So P here is the orientation of these like molecules that are embedded in the nematic. And we're only creating that polar order where there's deformation. So it's dark purple where you have high bend. We make up some rules about how the orientation should occur relative to Q. And then we borrow from electrostatics this idea of a polarization charge density, right? So you imagine that if you have a bunch of these dipoles next to each other and they're all aligned, then the heads and tails sort of cancel out everywhere in the bulk, but it's only at the edges of these polar regions where you have sort of net production of one, magenta winning or cyan winning. Okay. So I think this is entirely too fast, but here is the system sort of working. So on the left is this polar order for these enzymes that are embedded in the nematic. Purple regions are like where these dipoles are activated. And you can see that they're all kind of glued to the various defects, right? And so along the boundaries of these regions, you have high divergence of P, and that's giving you that effective polarization charge density, right? Where you're biasing the creation of one of those chemicals over the other. And as a result, those chemicals end up residing near the cores of their respective defects. Okay, and so we essentially now have a chemical label for our defects. In my flow diagram that I showed, that was only one piece of the problem, right? This was like learning how to make reaction diffusion couple to the mechanics. And then the next phase of the research is to think about, oh, well, now that I have this sort of toehold into the geometry of the system, I can now propose other kinds of chemistry. And I guess here's just sort of a cartoon to kind of reinforce what those continuum fields were trying to show us. This is how I envision these sort of dipoles being arranged in the nematic with the plus half and minus half defects. Okay, and so, happy to, explain any, much more of that if you have any questions. But the main takeaway is that we have chemical and geometric anisotropy kind of coming together to enable us to sort of label the topology of the system. And it seems kind of far out there, but I really think that when you look at the abilities of geometry sensing proteins that are already out there, and you think about that being combined with the fact that we are, I think, soon entering an age when we can design our own proteins, or DNA origami, we really have the ability to design these nano machines that enable these like higher level functionalities in these materials. So with that, I just want to thank all of my mentors and collaborators, and I'd be happy to take any questions.
Thank you, Mike. Do we have any other questions?
I'll I'll ask, just like for your numerical simulations and such,
Yeah.
what software are you using?
Yeah, for the ones that I showed, I'm using CalmSol, but it's like with a lot of regret, or it's like it was extremely challenging to set up the control problem in CalmSol. But the more recent stuff I've done in Phoenix,
Okay.
and I don't know if you've used that at all. It's an open source Python package.
Okay.
There's a grad student at Brandeis who's like redone all the control work, but using Phoenix. What I would really like to do is, I mentioned this like adjoint approach, right? Where you're doing this like you do a forward run with a simulation, then you do a backward run. There's a lot of new work out there that like borrows from the machine learning toolboxes to like automatically do that differentiation. So you don't need to do the back propagation. I haven't learned how to use those tools yet, but that's what I'm trying to do now. There's a lot of stuff in Julia, a lot of stuff in Python.
Cool.
So I can get away from CalmSol and its licenses.
Yeah, right. This is where, because it's like I know of ComSol, and then I'm like, just wondering if what other things are out there besides that. So yeah, cool. Thanks.
Yeah, thank you.
It made it sound like regret was a necessary condition to get ComSol to work. But yeah, it sounds really hard.
Yeah, I just think it with this like saving different solutions, right? Because it was just sort of a bear to like make it do this like looping thing. But
Well, anyone else? Okay, yeah, I think we're set. So okay, well, thank you so much, Mike, for taking your time and showing us your research. And yeah, we'll just thank the speaker one more time, and that'll be it.
all right, thanks a lot, Young Min. Thank you all for coming. Nice to meet you.
Take care.
All right, bye-bye.